Welcome to our second session on electrogram recording. I'm Sonny Jackman from Oklahoma City. My co-moderator is Pascal Van Dessel, who is from the Academic Medical Center in Amsterdam, and he's going to be presenting the first paper, Unipolar, Bipolar, Omnipolar, and Laplacian electrograms. Pascal.
Thank you very much. As always, it's a huge pleasure to be here, and I'm always especially excited because I think I always get the best topic of the entire summit, because what's more interesting than go back to real basics of electrophysiology, and that's the electrogram. So fortunately, I don't have any disclosures, so that saves us some time. And what I'm trying to do is talk to you about the genesis of the extracellular electrogram, the different modes of recording, and the stuff you can do with it in the clinical EP lab. So we're going back to a little bit of basics, and much of this was already covered this morning in the first session, and that's the way a cardiac action potential propagates across the myocardium. Here you can see two cells connected to each other, with the first cell is in the activated state, and of course the activation is caused by the fact that under normal circumstances there is a voltage difference between the inside of the cell and the outside of the cell, because the ion concentrations differ between the inside and the outside of the cell, and there are selective channel proteins that are selected for a specific ion carrier that can open, usually voltage gated, and allow a current flow into the cell. So the action potential starts, of course, with the opening of the sodium channels, and that will give the cell 1 a positive cell potential in contrast to cell 2. So there will be a voltage gradient between the cells, and at the sites where the cells are connected in the intercalated discs, as Dr. Kleber already said, there are connections which facilitate flow from the first cell to the second cell, giving membrane voltage elevation in the second cell, opening of the sodium channels, and propagation across the cells. And if you make there a very simplistic model for, and this is a myocardial bundle, like Dr. Kleber showed, all cells in a row, then you can depict the activation over this bundle if you stimulate here at the left side, and an activation front will be running across the bundle, where of course the leading edge of the activation front will be plus 20, because cells are there most recently activated, and the areas of the bundle where the wavefront has passed is already repolarized, so it's back at minus 90, and there's a gradient between those events. And so sort of you can picture it as an action potential that is reversed and traveling backwards across a bundle, and that shows the voltage gradients over a bundle. Well, if you take a little closer look at the leading edge of the wavefront, then, like I just said, the potential on the left will be positive, the potential on the right will be the resting cell membrane, potential of minus 90, so there's a voltage gradient. And the voltage gradient just at the site where the activation front has passed is not all that large, neither is it here at the site where the activation front is going to pass, but at the site of the steepest voltage gradient there is a large voltage gradient, and therefore also a large current. This means that the current will have to increase from a small current to a large current, and then again back again to a smaller current. So that means that voltage carriers will have to be recruited. And since the number of voltage carriers within the cell is limited, voltage carriers will have to be recruited from the outside of the cell, and therefore there will be a current across the membrane, a transmembrane current of voltage carriers going in and going out of the cell, and you get a current dipole that also runs across the extracellular matrix, and because this extracellular matrix has a resistivity, there will be an electrical potential field generated outside the cell. And this was very nicely mapped by Dr. Speck in the 70s of the last century, and he measured the strength of the potential field across the activation front, and he discovered that the potential field was positive in the ahead of the activation front, and it was negative at the trailing end of the activation front, and the magnitude of the potential field is largest the closer you get to the activation front, and at the exact site where the activation front passes, it will be switching from positive to negative and actually be zero. If you then want to know what that affects on the extracellular electrogram, you can position 3 electrodes along the myocardial bundle. And this is the electrode that is located near the stimulus site. And therefore, this electrode will only be positioned in the negative trailing field of the potential field, and therefore only see a negative deflection, which is largest when the activation front is close to the electrode, and which will gradually become zero as the activation front moves in this direction. The electrode located in the middle of the myocardial bundle will first see the activation front approaching, and therefore be located in the positive half of the potential field, and therefore initially see a gradually increasing positive amplitude. And at the moment the potential field passes the electrode, it will be located in the negative half of the potential field, and therefore there will be a quick switch to a negative large potential, then again become smaller again. And the electrode located at the end of the bundle will always only be located in the positive part of the potential field, and therefore only a positive deflection will be seen. So if you want to see that in a small animation, then you can see that if you stimulate this and you look at the traveling of the potential field across the bundle, then you can see the morphology of the electrograms appearing as the activation front spreads along the bundle. So how can we use this to determine activation times? Well, there are several methods, and we'll first concentrate on the unipolar and later on the bipolar electrograms. And if you want to record a unipolar electrogram, you connect your mapping catheter or your electrode of interest to the positive input of your amplifier, and you call that the different electrode, and the negative input of your amplifier will be connected to an indifferent electrode. And in theory, that will be an electrode located at infinite distance from the heart, but of course in practice that is a little bit difficult, so therefore you usually chose a remote site from the heart, for instance an electrode in the inferior caval vein or Wilson central terminal reconstructed from the service ECG. And then you get these kind of deflections that I show you that in the theoretical model of the first part of this presentation. And in the lab of Giel Janssen, they did the validation of where to place the activation moment in the unipolar signal, and they used intracellular recordings, and at the same location they made an extracellular electrogram, and indeed the upward stroke of the exopotential coincided with the most negative dVdt in the unipolar signal. And what even was more important, that if you created pathological conditions, for instance ischemia, then the exopotential is really distorted, and the unipolar electrogram is bizarre, but again, the principle that the most steep negative part of the unipolar electrogram coincides with the upstroke of the exopotential still holds through. So it's a quite robust method of determining the local activation time. Of course, you can also make a bipolar mode recording, and that has some advantages and some disadvantages. In this case, you connect again the different electrode to the positive input of your amplifier, and the indifferent electrode in this case is an electrode located in the heart, close by the different electrode. And what happens then is that if you have a wavefront that is passing the electrodes of your catheter, then in fact 2 unipolar electrograms are recorded, which are subtracted in the amplifier, yielding a bipolar electrogram. And in fact, what happens is that 2 unipolar electrograms with a little time shift are subtracted from each other, and if you put it in a mathematical formula, then in fact you subtract 2 signals with a little time shift, and if the distance between the electrodes is very small, then you can convert this to dVdt, which means that the bipolar electrogram is in fact the first derivative, time derivative of the unipolar signal. Well, why would you do that? Because unipolar and bipolar electrograms have significant advantages. The unipolar electrogram contains quite a lot of remote content because it can already see the wavefront coming from far away, and if you subtract that in a bipolar electrogram, then most of this remote content will be eliminated, and you get a clearer look at local activation. Please note that not all the remote content from the electrogram is removed, so there's still some significant remote content present in a bipolar signal. So if you, as unfortunately some people do, put your activation marker at the beginning of the bipolar signal, then you're not measuring the true local activation, because the true local activation, if that's the steepest DVDT in the unipolar signal, should be the largest either positive or negative amplitude in the bipolar signal. Remote deflections are not always misery, and I brought this one along with me because I thought it was a nice example. It's an example of a mapping done in an explanted human heart in a patient with a huge anterior myocardial infarction, and a multi-electrode balloon was placed in the left ventricle and VT was induced, and we were able to track the entire circuit of the VT. And you could see that it here exited from the scar at the septal site, then went around the left ventricular cavity and entered here the myocardial scar at the lateral side of this infarction. And you can see that very nicely in the signal. So here at A is the exit site where you have a steep descending unipolar signal. Then it goes through the viable myocardium where you get RS signals, and here at electrode G it goes into the scar, and there you can follow in the scar with minute but sharp deflections in the unipolar signal the tract of the arrhythmia. And the nice thing is, if you can see, if you look at the remote content at the signals obtained from the myocardial scar, then you see, of course, first here a remote R representing this remote activation at the beginning of the re-entry circuit, and it ends with a remote Q pattern indicating that you're already approaching the exit. So you can use, in this case, the remote deflection to judge where you are in the re-entry circuit. Well, there are a couple of disadvantages of bipolar recordings, and that's first of all that you should remember that it's very direction dependent. So if you have here again a bipolar recording device and you have an activation front that goes from left to right, then you get this nice bipolar signal where the local activation is determined by the, in this case, largest negative deflection. If you have exactly the same wavefront and now it's coming from right to left, then you get a mirror image of this bipolar signal, and in this case the local activation is the largest deflection. Unfortunately, if you have again exactly the same activation front but it's coming perpendicular to the electrodes, then both same signals are subtracted and you will see nothing, so it will appear as if there is no activation at all. Now, of course, the distance between your poles is also important. As I told you, the distance should be very small, and if that's very small, then you get a true bipolar signal. If the distance between the electrodes is very large, then you get a sort of a weird signal of two unipolar signals with one upside down, and I really wouldn't know what to do with this. Of course, also the orientation of your bipole towards the myocardium is important. If they are both nice in contact with myocardium, then you're subtracting two true unipolar electrograms, and you get a bipolar electrogram. If your catheter is perpendicular to the myocardium, then one will be recording a cavity potential, the other unipolar signal, and in fact what you think is a bipolar signal is sort of a unipolar signal actually. Main advantage of bipolar recordings is that it's less sensitive to noise because the electrodes are located close together. So if you, for instance, have 50 or 60 hertz interference from the main, then probably the noise will be the same on both electrodes, and if you subtract it, then you still get a clean bipolar signal. If you're using a unipolar signal, since the other electrode is located far away, then probably the noise will be different, and if you subtract that from each other, then you still keep a noisy signal. Well, how do you do that best in practice? So here you see an example of a mapping we did in the RVOT of a patient with Brugada syndrome. You see a complex unipolar electrogram with multiple deflection, and this one is clearly the sharpest, so we call this the local activation. And if you look at the bipolar electrogram, there are also more deflections, and in this case not the largest positive deflection is local activation, but actually the largest negative deflection. Well, a third mode of recording is the Laplacian mode, and in this case you add another electrode, and they are equidistant from the different electrode, and you subtract the corrected for the number of electrodes, the electrograms from the different electrode, and in that case, you get sort of two bipolar electrograms that you are subtracting against, so you get the second time derivative of the unipolar signal. And the nice thing is that you even have less remote content, and that the unipolar morphology is sort of restored in the Laplacian electrogram, so you can again make more deductions on the morphology of the electrogram. Well, how can you use that in practice? Here you see mappings in a dog with ventricular fibrillation. There you're using a multi-electrode array, and here you see the unipolar electrogram for the electrode in the center, and here you see the reconstructed Laplacian electrogram by subtracting 4 of its neighbors, and you can see that it's much easier to determine the local moment of activation, and you can also see that the waveform information is kept, because here you see only an R wave, so that means that the activation front stops at this point at this electrode, and in all the other electrodes it will be continuing. And this I'm going to skip because of the time, because there's a dedicated session on this afternoon, and unfortunately I'll have to skip this as well, so I'll stick to the caveats that you should use when you're mapping. And here I have made a sort of a thought experiment. We measured a virtual arrhythmia with an electrode grid with 1 centimeter electrode distance, and we determined correctly all the activation times, and you get a pattern of a focal activation with centrifugal spread across the myocardium, so you would be inclined to take your catheter and start ablating here. Well, what happens if you increase the number of electrodes on the same arrhythmia? Now you can see you get more information, you can measure more activation times, and in fact this activation pattern was caused by a figure of 8 re-entry with a zone of slow conduction that was not covered here in this area, and therefore a totally different mechanism, and it should advise you to start your ablation in this area. Well, if you want to calculate conduction velocity to the electrodes, then it's tempting to assume that the wavefront is going straight from this electrode to that electrode, and when the distance is 0.6 millimeter and the time distance is 5 milliseconds, then you come to very slow conduction. Add some more electrodes, and it can turn out to be that in fact the activation front is forced by local barriers to follow a very complex activation pattern, and that in fact the conduction velocity is normal, but the way between the electrodes is much longer. And my third suggestion is that you always should pay attention to the sampling rate of your signal, of course. You see here electrograms obtained from the junction between the Purkinje system and the ventricular myocardium. And here you see very low sampling rate, and there's no Purkinje spike whatsoever to see. And only from at least 1 kilohertz you can see a Purkinje spike starting to arrive. And of course you see it best at 10 kilohertz. Just always be aware that your sampling rate is adequate for the type of rhythm you're trying to map. So what was I trying to tell you? We know now how the extracellular electrogram is generated. There are several methods of how you can measure the local electrogram, and each has its own merits and its own drawbacks. The action potential surrogate, unfortunately, has to go because I had 20 minutes last year and only 50 minutes this year. And when you're looking at, of course, our business has improved immensely by sophisticated mapping tools, but always try and understand the underlying electrograms, because a cartoon made by a mapping system is only a model of reality and not necessarily the reality itself. And if possible, always look at bordering electrograms to analyze the electrogram under the electrode. Thank you.
Well, Pascal, you covered so much in just a couple of minutes. My topic, and I want to thank, I don't know where Alad is. I can't see that far. I have the wrong glasses on, but thank you for the invitation. It's an amazing program, and I'm honored to be here. So my topic was challenges in the accurate measurement of activation times, which is a beautiful follow-up, I think, to Pascal's talk, and it'll help a lot. So remember what Pascal said. So with a conventional electrode, we measure the activation time from the unipolar electrogram, and you remember, as he showed, as the wavefront crosses the electrode, you go from the most positive to the most negative voltage that that electrode is going to see. So you go from the most positive to the most negative. So we measure the activation time at that steepest negative slope of that downstroke, and as Pascal mentioned, the systems, the mapping systems that we use that measure the activation time, they use the derivative of this curve, and the steepest downslope is going to be the most negative point on the derivative curve. So normally, in normal myocardium, this is really quite easy. You have a single wavefront coming across like that. It's very easy. When you get into diseased tissue, heavily scarred tissue, now you're looking at multiple negative, downslopes in there. So the question is, which one do you select for the local activation time? So as Pascal said, by looking at the bipolar electrogram, we tend to eliminate or minimize far-field components. I should have mentioned that when you have a bunch of components in our electrograms, it's because you're seeing a lot of things all at once. You're seeing the actual activation time, you're seeing activation times that are occurring nearby, and you're seeing some far-field activation times. So as Pascal said, we can use the bipolar electrogram. We can use the bipolar electrogram to minimize or get rid of the far-field components. So we always start with the bipolar electrogram, and you're looking for the sharpest components. So the local electrogram has to be a sharp component. To my eye, the sharpest component is here, but there's somewhat sharp component here, and who knows, maybe a sharp component at this point. But if you look at this area here, everything is rounded. It's not sharp at all. So that's far field. If you look at this area here, these components are also rounded. They're far field. You can eliminate those from the evaluation. So what that does is that limits the possibility for the local activation time to be one of these 3 times. So you can kind of drop an imaginary line down from the sharp components of the bipolar electrogram, look at your unipolar electrograms, and you see that this downstroke here is relatively sharp. This one is a little bit less sharp, this one less still. So if you were to just eyeball this, which we don't use on our systems, but as we're looking at electrograms ourselves, you'd say, well, that looks like the local activation time. And in fact, the mapping system did select that. The steepest, the most negative derivative of the unipolar electrogram was at that site. Well, what if we wanted to measure the activation time of electrode 2? Well, we're going to use the same bipolar electrogram because it includes both unipolar 1 and unipolar 2 in this bipolar electrogram. So it's got to be one of these 3 sites. And we look at this one here, this first one, and it's pretty sharp. There's nothing here at this second one, and there's really no significant downslope on this third one. So this one's a slam dunk, right? This would be the local activation time of the second electrode. But what if you said, wait a second, Sonny, I mean, you go to all this trouble looking, you're going to wind up on the unipolar electrogram. Why not just look at the unipolar electrogram and analyze that? Well, let's do that. Let's just look at the unipolar electrogram and let's blow it up. So if we blow it up, you can see that the steepest downslope is right here. And where that is on the electrogram is right here. And if we look at what we have on the bipolar electrogram, it's pretty rounded there. That makes this point right here actually a far field potential. You wouldn't want to take that. So the summary of this component of the talk would be that you always want to look at the bipolar electrogram first, pick out what the possible local components are, and eliminate all the far field areas. And then you can pick the steepest slope and take that as your starting point. One of the things I want to mention, I want to come back to this, and I want to finish on this point, and that is if you look at scars, or if you look at diseased areas, heavily scarred regions, very arrhythmogenic regions, almost always the largest, sharpest signal on the unipolar electrogram is far field. And the reason is, traditionally, we have used the Wilson Central Terminal for the indifferent electrode that Pascal was talking about. So if you look at the distance between the electrode that you're actually measuring, what he referred to as the input on the differential amplifier, called the different electrode, and the reference electrode, they're very far apart. So that distance gives your unipolar electrogram a huge field of view. It sees a large area. And so it's very easy to pick up far field. And in diseased areas, the voltages are low. And in the surrounding area, the voltages are very large. And so you typically get the largest, sharpest unipolar electrograms are far field. And the problem is there's sometimes a little component on the bipolar. And so one of the biggest problems we have in mapping is interpreting far field signals as local. Now, one way to get rid of this is to bring the indifferent electrode or the reference electrode much closer to the recording electrode in that unipolar electrogram. So you have a close electrode, but non-contact. Now, when the wave goes by, you're going to have a normal unipolar electrogram from the measuring electrode, the electrode you're measuring the unipolar electrogram on. If you look at the recording that you're going to get from the second electrode that you're using as your reference, the activation time is going to be the same, but this electrode is far away from the tissue, and so the amplitude and especially the slope is going to be markedly reduced. So when the amplifier subtracts this from this, you wind up getting an electrogram, and Pascal said it, you said it very, very quickly, but you introduced it beautifully for me. You get an electrogram that looks like a unipolar electrogram. You get a bipolar electrogram that looks like, in times, like a unipolar electrogram. It has some added value. The first is, when the wavefront is far away, you won't see anything, right? You have to get close because the electrodes are closer together, so the field of view that the electrode has is much smaller. The wavefront has to get a lot closer, so a term I use in my head, I don't think there is such a term, the recording range, how far the electrode sees, right, is much, much smaller. Okay, and you're going to get that anytime the reference that you use is close but not in contact. And about 18 years ago, I remember when I was working for a little while in Amsterdam, Jacques de Bakker, I heard him refer to this as a close unipolar electrogram, meaning you get the advantage of eliminating far field potentials. Now, this is going to have another advantage, and Pascal referred to this also, and that is it eliminates the 50 to 60 hertz noise that you get with unipolar electrograms when you use the Wilson Central Terminal. Why do you get that noise? Or why do you eliminate it? Well, you eliminate it because now both electrodes have the same impedance for the amplifier. That's one good point. But more importantly, the wires travel together to the amplifier, so you get common mode rejection. And that's the reason you get rid of the noise, and you don't have to use bad amplifiers. That drives me crazy. Everybody uses the notch filters, and I think they're like the biggest enemy that we have. And I don't ever use them. Now, what I'd like to do is I want to show you an example. I want to finish out by showing you an example of a recording that has both a close unipolar electrogram and the same electrogram recorded using the Wilson Central Terminal. And by the way, I guess I meant to tell you that I think that using a close unipolar is a far better way to get your activation times, much, much more accurate. And you're going to have that anytime the electrode is close but not in contact. So to give you a comparison, to show you a simultaneous comparison, I want to just mention that Biosense Webster has developed 3 different types of multi-electrode catheters to take advantage of a close but non-contact electrode for reference. So their unipolar electrograms are between the recording electrode and a non-contact electrode. And this particular one, this mini basket electrode, is one where the preclinical study was done with a system that allowed simultaneous recording of both of these, and that's why I want to show that to you. But I'll just show you this catheter. It's a mini basket catheter that has 10 splines with 10 tiny outward-facing electrodes to it. And the preclinical model that was used is a canine right atrial model, where an RF lesion, a linear RF lesion, was created between the superior vena cava and the inferior vena cava to get some abnormal electrograms, sometimes to leave a gap, and it's kind of a lovely sort of model, and pacing from the low anterior side of that line. So here is the uncensored or unmonitored or unmodified activation map. So this is just how it came out of the machine using the Wilson Central Terminal. And I just want to look at one electrogram right here that's in the middle of the ablation line and is right here on the map. So here is the bipolar electrogram that was recorded. And you see the atrial stimulus artifact is right here. You can't see the red line, which is the reference timing. Here's the window for the mapping system, and you can see a long fractionated electrogram, multiple components. The first component is a little bit larger at about 0.5 millivolt, but all the rest of these are all less than peak to peak, all less than 0.05 millivolts. So pretty small in our world. And as I look at this, you say, well, which one of these is the local activation time? If I was looking at the bipolar electrogram saying which looks steepest, this is the one that, I would pick. Remember, we have to look at the unipolar electrograms, but I would start by looking at this one. I would think these two might also be candidates, but let's go ahead and add the unipolar electrograms. So here are the unipolar electrograms measured using the Wilson Central Terminal. So between electrode 2 and the Wilson Central Terminal for this unipolar electrogram, and this one is between electrode 3 and the Wilson Central Terminal. And here are the electrograms. So CARDO, for every bipole, it actually annotates or takes the activation time of the distal of the two electrodes. So we'll look here. And it turns out that the steepest downslope, or the steepest negative, or the most negative DVDT, or the steepest downslope, was actually all along here, was actually right here, and that's at 41 milliseconds from the reference time. Okay, I want to now show you the unipolar electrograms recorded with the close non-contact electrodes. So I want to pull this up to make some room here. And these are simultaneously recorded. I put a white band here just to make it easy for you to see. This is the Wilson Central Terminal. These are the same two unipolar electrograms recorded using the close unipolar electrogram. And I want to point out a couple of things. The first thing is the close unipolar electrograms on the far field components, the amplitude and the slope are markedly reduced. And just to point this out, you can take the biggest far field, that's definitely far field, right? And that's the ventricular electrogram. Obviously, we're not in the ventricle. Oh, and I forgot to mention, I apologize for this, it was on the previous slide. These slides were given to me by Hiroshi Nakagawa and Asaf Govari. And I meant to mention that earlier. I apologize. Okay, so back to the talk. The biggest far field, or I should say the truest far field, or what's really far field, is the ventricular signal. And look at the size and the slope of the electrogram here. Now compare it on the close unipolar electrogram. You can see how it's markedly reduced, but notice the slope is a lot less. Okay, all right, let's look at some of the atrial ones. So we'll look at this one here, which was the steepest, and look at the change. Look how much lower the amplitude, but more importantly, the slope is less. All right, let's go to this next one here, okay? And it's almost completely diminished. It's almost completely diminished, and it's clearly far field, okay, as you look at the bipolar electrogram. What about this little one that I wondered about out here? you can see the slope here is also less. And in fact, the polarity is normalized and made correct here. It's flipped here. So the key is the close unipolar electrograms reduce the slope of the far field. Interestingly, they accentuate or steepen the slope of the local components. So here was the steepest slope. Here's the steepest slope, and when you look at this here, okay, compare it to this one. It's actually larger, but that's not the key. The key is that the slope is steeper. And remember, we're going to annotate, or the system annotated C2, and when you look at the most negative derivative of that, it's located right here. And so instead of the timing being 41 milliseconds after the reference, it's 118 milliseconds after the reference. And so you can see the importance of being able to differentiate what's truly local by getting rid of the far field, by bringing that electrode close. So on the map, here was the bipolar map. Here are the unedited, that was the word I was looking for before. I had a senior moment there, you know, as I was speaking. These are unedited, so this is the close unipolar electrogram map. So this is the map made using the timing from the close unipolar electrograms, and here is the Wilson Central Terminal. This was the point that we compared. Now, in all of the swine in this study, the crista created a line of block in all of these. And then you can see the line of block from the ablation line here. And you can see this error point here. There's also a lot of other error points. And you can see it makes it a lot harder to actually see the very discrete line. And that was also true for gaps in the line and whatever. Here's another thing. The Accardo was unable to identify an activation time for these gray points here. No problem at the same time seeing it with the close unipolar electrogram. And so the clinical studies are beginning to bear out that using this close unipolar electrogram markedly tightens or improves the accuracy of this. And I think we're going to see in the future that this is going to be a great improvement for our mapping systems. Thank you very much. And I'm going to go ahead and introduce our next speaker. And our next speaker is Edward Vigman. And Dr. Vigman is the head of the modeling group at Lyric and University Bordeaux. And your topic is translating conduction velocity measurements from 2D to 3D.
Yep, thank you.
Thank you, Edward.
Very nicely following up on your talk. I'm going to assume that we have the proper annotation and that we have a proper identification of the activation times. So why do we want to know what the conduction velocity is? Because it can be an important factor in arrhythmia development. Very important to know the conduction velocity times the APD gives the wavelength, which is a measure of how many rotors or scroll waves you can fit on a surface. This, as we heard earlier this morning, slow conduction velocity might be related to either change in safety factor or not. And unfortunately, conduction occurs in the heart as a 3D structure, but we can only see what's on the surface. And we have technologies which are improving to measure CV, but what are they measuring and how can we use what they measure? Okay, so the way I'm going to measure CV conduction velocity, I use meshes composed of triangular or tetrahedral elements. So assume we have three corners of our triangle, and these are the activation times at each of the corners. And we fit a linear function over this that describes the activation time. We take the gradient of that, and then we can just take the inverse of the magnitude, and that gives us the conduction velocity. And we're going to do this for different resolutions, for different sampling. So we're just going to take my original mesh, it was a 0.5 millimeter resolution, and we can downsample it to any resolution that we want. Also in my model, we're going to see a Purkinje system. And what's the Purkinje system look like? we use a model, we generate that using a fractal sort of pattern that covers the endocardial surface. You'll notice that it sort of looks like little hexagons, and it turns out hexagons are the most efficient method for covering a surface. You use the least material to cover a surface using hexagons than any other surface. And it has a 3D structure, and you get quite a different activation. Normal this is a simulation of normal sinus activation on the right. So you can see the complexity we heard about in the first from Dr. Kleber talking about showing Durer's map. It pretty well follows. You have activation is actually mostly transmural initially, and then towards the base becomes transverse. Okay, so one of the problems, first of all, with 3D is measuring on the surface, is that the wavefront is moving in 3 dimensions. So for example, we have the endocardium, which is the dark red line. We have the wavefront blue, which is moving in the direction indicated by that black arrow. And we have an angle alpha between the wavefront and the endocardium. So in the same amount of time that a point on the surface we're going to see move from the left to the right, the true conduction velocity is going to be moving in the tissue, is moving from this point where they're going to intersect. And as a result, the conduction velocity you see on the surface is the conduction velocity you see in the tissue divided by the sine of that angle alpha. And sorry for the translation from Keynote to PowerPoint, but you can easily get a value of 0 if this alpha angle is 0, which means that the wave simultaneously hits the surface totally parallel to it, and you assume. So even this matters, even normal propagation. So for example, if this is a slice of tissue at the epicardium and the endocardium, and both of these surfaces sort of have a liquid interface, whether it be blood or whether it be some fluid between the pericardium, and this is there's a bath loading effect, which means that the conduction velocity is slightly faster on the surfaces than it is in the middle, and as a result, to account for this difference in tissue and surface conduction velocity, the wavefront actually has to travel at a slight angle to the surface. So this is even in normal undiseased myocardium. Another issue with measuring the 3D surface velocity is breakthrough sites. So here's an example of activation. We're looking at the endocardium, and we're looking at it. There's a Purkinje system that's responsible for this activation. I'll play it again. And you see, and so if we look at the activation map, and if we look specifically at a breakthrough site, we see this triangle. So if we were using my previous algorithm to determine the conduction velocity, we can see the corners are 34, 35, and 36 milliseconds. So it seems like it only took 1 millisecond to traverse this element. But really, activation started in the middle earlier and took almost 2 milliseconds to go there and 1 millisecond to go there. So this should really be more like 3 milliseconds to go across the bottom. But when you do the calculation, you're going to get a spuriously large conduction velocity. And how much does the Purkinje system affect this as well? Because we never have pure myocardium. So this is some experimental work done on a pig, and this is the right ventricle, and was experimentalist at Lyric, David Benoit, and he takes a right ventricular wedge, and he either stimulates at the RVOT or at the RV free wall at the bottom, endo or epi. And when he does his experiments, he gets a different time to activate the endocardial or the epicardial surface based upon where he stimulates. So whether he stimulates at the RV endo or epi or the RVOT, the time for the activation of the epicardium or the endocardium changes. So when I first took a stab at modeling this, I didn't put a Purkinje system, I just put a slab in, and then I got a result that was both too long and did not vary upon where I stimulated it. And I was using, again, we never just talked about this yet, but the heart has 3 directions. There's a fiber direction, a cross fiber direction, so that's parallel to the plane, the endocardium, epicardium, and then there's a third direction, which is the normal going from epi to endocardium, and each of those directions has a different conduction velocity. So I used it 0.6 along the fiber, 0.3 across the fiber, but in the plane, and then half of that, 0.15 meters per second, going from endocardium to epicardium. And if I added a Purkinje system, then I was able to recreate very well the characteristics of the experimental recordings. So it isn't always obvious that the Purkinje is playing effect, because sometimes the breakthroughs are so close together, they merge together and you don't see distinct breakthroughs. But nonetheless, there's still Purkinje activation, which is increasing the conduction velocity. So another effect which might actually affect conduction velocity on the surface, which doesn't have anything to do with the intrinsic conduction velocity of the tissue, is curvature. So here is an example of a simple activation time in a 3D ventricle, and the blue line is a wavefront, and then you have at each point on the wavefront, you can figure out the normal direction. And then the red lines are the fibers, so those are the predominant directions of the myocytes at that point. And it's actually the conductivity that the wavefront sees depends on the direction of the normal relative to the fiber. So the current flows easily along the fiber, so on that direction, the conductivity is the lowest and the conduction velocity is the fastest. So we can use the conduction velocity, this conductivity, to figure out our conduction velocity. But curvature is actually not based upon the wavefront normal in this condition. You have to modify it by the fiber direction. So you actually get slightly different normals, which are the green arrows. And it's those normals you have to use to compute the curvature. And if you look at just normal propagation, just from starting at the star, you can see this distribution of curvature in the 3D ventricles. And how does curvature affect conduction velocity? It's pretty much a linear dependence. This has been well established. And you can see it can have a significant range on the conduction velocity going from, here I have the graph from 700 to 550 over curvature from minus 0.6 to 0.6 per millimeter would be the, and here you can get higher values than that within this structure. So to look at the effect of curvature and conductivity on propagation velocity, I took the curvature map, which So to explain this a bit, so on the left figure, this is the conduction velocity that was measured in the simulation. So we took the activation times and the conduction velocity. For this middle conduction velocity, I took the activation map. Then at each instant in time, I figured out the normal direction, which lets me figure out the conductivity. And then strictly using that conductivity, I computed the conduction velocity. And I got this map, which is a pretty good. And then as a final refinement, I took the curvature effect, added it to the conductivity effect, and produced it. And for the most part, the conductivity directionality explains probably 95% of the conduction velocity. There's a few regions of high curvature, which I've highlighted here, and you can see slightly different, or maybe it doesn't show up so well, the change on the conduction velocity. So let's get back to what happens when we measure something on 2D. So I took, there's a biventricular model that I have, and without a Purkinje system in there. And the gold outline, the gold curve there you see is the actual 3D conduction velocity. So, and then the other curves are the conduction velocities computed from just surface values, from just the endocardial values. So you can see there's quite a big difference between because the surface cannot track any out of plane direction. If we add the Purkinje system, then we see that actually we get a much these are all the same scale, so you can see that you actually get a lot of slower true conduction, and that is explained by when you have Purkinje system, you have a lot more transmural conduction because you're activating the entire endocardium at once, so the propagation has to occur from endo to epi, which is the slowest direction because you're crossing like the heart is an onion, you're crossing all the sheets in the onion, and that's the slowest direction. And you see you get spuriously large conduction velocities when we calculate these along the surface. We change the pacing location more to the RV apex. Again, we get spuriously large, and for really large mesh resolutions at like 0.2 and 4 millimeter spacing, we start to get spurious low conduction velocities. And sinus rhythm is probably the hardest thing to capture because it's, and there's not a very good agreement between the true and what's measured on the surface. So again, there's regional differences depending on where we look at the conduction velocities that we measure, whether this is the intrinsic. But generally, the trend is no matter where we are, what we measure on our surface velocity is going to be is generally too high. Okay, and activation, this shows the same thing. So the left is if we do His bundle normal activation, so the red line. So on the y-axis is the 2D conduction velocity, on the x-axis is the true 3D conduction velocity. And if we look at when we do the RV apex pacing, the conduction velocity is consistently higher than the true because it's above the identity line. But what happens, but maybe close to the electrode, maybe there's some hope in there, maybe it's not such a bleak story after all. So if if we pace at the star and those lines, those are isodistance lines, they're spaced 1 centimeter in space from the stimulus site. And if we look at the correlation between the true, and we see this is the error, so we're plotting the error in the surface conduction velocity versus the true conduction velocity. And we see actually it doesn't do such a bad job. When we don't have a Purkinje system, the error is less than 10 centimeters per second within 4 centimeters from the pacing electrode. So once we do add the Purkinje system, then of course it's only more valid for within 2 centimeters, using the same error threshold. And that has to do with, of course, with the Purkinje system, you're getting less out of it's no longer, the more the role the Purkinje system plays, the less role the intrinsic myocardial conduction matters, because propagation is being carried primarily through the Purkinje and then being transferred into the myocardium. So in regions where you have less Purkinje, you're going to get probably truer to the conduction velocity that you want, because you want to know the myocardial intrinsic conduction velocity. We did this in pigs as well. We did pig experiments using plunge needles, putting them through the wall of a piece that's in the LV. And again, we noticed the same thing. The true conduction velocity, the 2D surface conduction velocities that were computed tended to be higher and would be spuriously large than what was measured experimentally. If you look at the direction, of course, as you would expect, the 3D one, the 2D surface totally ignores the out-of-surface components. On the bottom, we have the true one. The top is the surface. You can see all these surface. You see more arrows on the top because all the arrows are going along the surface, whereas in the bottom, the arrows you don't see are pointing inward. And if we look in detail, especially on the septum here, so you see activity crosses the septum from the LV to the RV. The conduction is pointed that way, and it's all on the inside. But here, when we do the 2D, you see it's wrong because it's constricted to the surface. The region of slow conduction. So yes, so we put in a region, as you might want to detect if there's a scar or fibrosis or what have you causing slow conduction. You can see this is without the region of slow conduction. As you would expect, you get the bunching of isochrones. And we see that we do indeed get the reduction in conduction velocity that we expect. And if we add the Purkinje system, again, this message is not as clear at all. But there does seem to be something. So intuitively, slow conduction does lead to more isochrones, but conduction velocity is a derivative of the activation map. So even though the activation map looks fine, you take a derivative of it, and it's much messier. It's just a mathematical fact that differentiating creates noise. And also the source depth has a certain effect on it. So again, things that are deeper in the tissue, like this, are going to produce much more different, this is the activation pattern if something is close to the surface. If the source of the activity is deep within the tissue, you get a much liquor, and these are compared, this is experiment compared to the real. So in conclusion, generally this conduction velocity you measure using surface-only measurements overestimates the true tissue conduction velocity. Purkinje involvement can play a role. There's a delay going into the Purkinje system and out of the Purkinje system. So the larger the distances, the more relevant the role of the Purkinje. The electrode spacing should be less than 2 millimeters. The 2D CV correlates well within the 3D as long as you're close to the stimulating electrode, within about 2 centimeters. And close to the electrode curvature could play an important role. And finally, conduction velocity, again, is a differencing operation, which means it's always susceptible to noise. And inverse solutions may be possible, but they would require a high-density mapping. And this is my collaborators from Toronto who helped me with this. So thank you.
Thank you, Edward. Do you want to join us? Our next speaker is Omer Berenfeld, who's a professor of medicine and bioengineering at the University of Michigan in Ann Arbor. And Omer's title is Methodological Considerations in Mapping Fibrillatory Rhythms.
Thank you, Dr. Jackman. Thank you, the organizer, for inviting me. It's a really great honor and pleasure being here. So I'll be focusing within the very large topic that I was assigned to on mostly optical signal processing, because others will talk a lot about electrograms. I will show some mapping computational approaches, and as time permits, I'll talk about driver and onset of AF as an implementation of the computational approaches. So this is a slide just to establish the fact that optical mapping is considered the groundbreaking, the gold standard mapping method. We can see here the similarity between the electrical recordings with microelectrodes and the fluorescence signal. The similarity basically establishes that this is indeed the best approach to map large areas of electrical impulses. The differences between the electrograms and the fluorescence is in the abstract. The fluorescence is slower. But nevertheless, we can obtain optically large heterogeneity in the action potentials from different regions. This is an example of one of from our ship experiments, illustrating that atrial fibrillation is in many cases driven by reentron activity, as seen here. But the rotors, the reentron activity is not easy to study. We know that in the center of the core of the reentry, the amplitude is very small, and the rotors tend to drift, not to be stable most of the time. So we enhance the characterization of our raw data, the movies. If we're interested in stationary properties, we will apply Fourier transform to determine the dominant frequency. And for dynamic propagation properties, we utilize the Hilbert transform to generate phase maps. I'll briefly go to see what they are. So in the frequency to the stationary properties, here is a movie. Each pixel is a time series. We apply the Fourier transform to determine the frequency at which the power is maximal, and we reassign that frequency as the dominant frequency. Then we color-coded it to get the dominant frequency maps. As for the dynamic properties, as I said, we utilize the Hilbert transform, and here I illustrate how that works. The action potential as the form is involved, but we could color-code it based on the phase. Here's the resting, upstroke, and so on. And the advantage is that no matter what the morphology of the action potential, even if the amplitude is very small, we get consistent descriptions of the, for example, the upstroke here in green. And that helps us illustrate propagation in one snapshot. For example, here we see that the green will propagate towards the blue, so we interpret that map as a reentron activity around this point in which phase is not determined, and we therefore call it phase singularity. Here's how we combine frequency and phase movies, phase frequency analysis. This is a movie of atrial fibrillation. The colors indicate phases. This is from the posterior left atrium. This is from left atrial appendage. But we have changes in the patterns. Here we can see reentron activity, and sometimes we have breakthrough activity. What we notice is that when the rotor is located in the left atrial activity, left atrial appendage, its dominant frequency was the highest. The difference with the posterior left atrium was not very large, but was very consistent. However, when the rotor abandoned the left atrial appendage, the frequency there dropped. So at this point, let's take a pause a little bit, and before we move forward, I would like to consider a few aspects about the data quality and primarily sources of possible inaccuracies. As for the dominant frequency, when we do that calculation, we always have to remember that harmonics or subharmonics in the power spectra can mask the true activation rate, which is the parameter we are interested in. And the duration of time series affects spectral resolution. That's important when differences are very small. Duration of time series affects also physiological interpretation of stationarity. As for the phase mapping, the Hilbert transform sources of inaccuracies, it is a very sensitive method for small amplitude variations in time series, and that may create false phase singularity points. And also importantly, we have to consider the fact that the Hilbert transform is a non-causality transform that may affect the phase, particularly at the edge of the time series. So maybe a few practices that we can use to reduce those accuracies. As for the stage of data acquisition, it's a very general good practice. It seems obvious. We have to increase reliability always for local activation time. When possible, of course, we use optical mapping. Obviously, that's not always the case. But great attention should be devoted to increase reliability. And we heard about that previously, and we'll hear more. When you do use optical mapping, we have to, as a moment, we have to abolish motion or account for motion, which distorts interpretation. And we obviously have to avoid spatiotemporal non-uniformities in illumination and fluorescence. As for electrical recordings, we have to stabilize contact to keep uniform the input impedance and avoid frequency-dependent biasing hardware filtering, which is sometimes overseen. As for the data pre-processing, what we do is, first of all, we do linear detrending, an additional about 1 Hertz non-biased high-pass filtering and zero tapering of the edges. We also use non-biased pass filtering for typical activation rates. In the humans, that's between about 3 and 15 Hertz. We apply rectification to reduce electrocardiogram variability. And in addition, sometimes we use non-biased bandpass filtering to specific activation rate, for example, the maximal dominant frequency. It's always a good idea to compare your results, no matter what method you use, with, of course, the physiological model you have, and also with subsample with time domain data. So having that in mind, the objective of the rest of my talk is a combination of time, phase, and frequency domain analysis to study rotors and acceleration of the atria during dynamic atrial fibrillation onset. In particular, I would like to look at non-stationary and stationary patterns during the initiation of the AF and to identify rotors that contribute to AF acceleration and sustenance. The methods I will talk about include new panoramic optical mapping of the endocardial of a sheep heart to study the acute atrial fibrillation onset. And here's the diagram of our setup. We isolate the heart and we insert 2 borescope with panoramic lenses, objective lenses, to study the activity in the entire 2 atria. This is a picture of the system, and here we can see the atria from inside. We can appreciate that it really looks at all the anatomical components of the atria. Here's our protocol. The experiments were done in the presence of 0.25 micromolar carbachol, so in a sense it's a vagal atrial fibrillation. Here we see pacing with the 10 stimuli in the left atrium and premature S2 that was followed by a relatively organized stage, which we call initiation, and it's followed by less organized activity and faster, which we term early stabilization. This movie is to show you, you can appreciate the quality of the signal. This is the pacing going from left to right, and initiation is self-sustained. And during early activation, early stabilization, we have faster activity and more complex activity. The complexity is also evident by the dominant frequency analysis. We can see that at initiation between 0 and 3 seconds, there's a large domain at 8 Hertz, and those dominant frequency domains break apart, become more smaller and faster when you go to early stabilization between 30 and 33 seconds. This is illustrated also here. We have from initiation between 0 and 3 all the way up to 30 seconds an increase in dominant frequency, and thereafter a stabilization. So the question that we're asking is how those dominant frequencies stabilize. And to answer that, we track cycle-by-cycle rotor analysis. We look at the core, and we look at the cycle length at the core. And then we plot those cycle lengths as a function of the cycle previous to the appearance of the rotor and thereafter. And here's the data from a collection of hundreds of rotors that we see everywhere in the atria. So what we see is that during initiation, we have a series of rotors, on average about 2 rotors per second. And before the appearance of the rotors, there seems to be fluctuations, but not necessarily trend in acceleration or deceleration. Just at the moment of the appearance of the rotor, we see a prolongation of cycle length, and that is preceded by abrupt reduction in cycle length, acceleration of the activity at the core size. Thereafter, the rotors don't last for many cycles, and after few rotations, they disappear, and activity may go back to the original activation rate before or accelerate. Interestingly, during early stabilization, about 30 seconds later, the number of rotors did not increase significantly, still about 2 per second. However, the pre-rotor cycle length is about 10% lower than those during initiation. Therefore, there was a general acceleration of the AF. But still, at every moment, the time the rotor appears, there is an abrupt reduction in cycle length. Thereafter, the rotors last for a few cycle lengths, disappear, and as we can see in some cases, cycle lengths went back to the pre-rotor cycle lengths. We look at that, well, we ask the following question. At the rotor cores, that the data I showed so far, activity always accelerates when they appear, but it may decelerate once the rotors disappear. So the question is, what is the activation rate across the entire atria, which is what matters more AF, when the rotors disappear? And the other question is, if rotor cores accelerate during initiation and early stabilization, I showed you accelerate in both situations, then what stabilizes the AF rate at early stabilization stage? So to answer that, we look at cycle by cycle length at the entire atria, not necessarily at the core. Here we have the rotor. During this period of time, we look at the cycle length before the rotor and after the rotor, but in the entire atria. As illustrated in this map, the cycle length map, there was in this case acceleration from 110 milliseconds, which is the smallest cycle length in the entire atria, to 67 milliseconds, which is the shortest in the entire atria, not necessarily at the rotor's place. But that was not always the case. We not always have acceleration. Sometimes we have decelerations. Again, this is the rotor time where the rotor is present. Here we have a cycle length before and after, and we look at the map of the entire atria, and we can see here, I don't see very well the numbers, but the number went from 63 before the rotor to about 67 milliseconds after the rotor. So there is a deceleration of the atria, although the rotor accelerated, the rotor itself accelerated. So we look at it more carefully, what is happening here. We collected during initiation 210 rotors from 20 episodes of onset of AF, and we notice that there is huge variability. I'm showing you individual rotors. In red, you can see those, for example, this one that created acceleration. The post-rotor cycle length is shorter than the pre-rotor. But there are many others in which there was deceleration, or at least no acceleration, the blue ones. Overall, there is net effect of acceleration from about 90 milliseconds to 87 milliseconds. Just tiny, tiny change in cycle length. But when you have many, many rotors, those accumulate, and the difference was significant. During early stabilization, still many rotors, 200 and few, you can see from here. There is again, some rotors accelerate the atria, others do not accelerate. However, in contrast to the cycle length at initiation, this time there was no net acceleration, which can explain why the atrial fibrillation stabilized its frequency. Here's a summary of those results, different summary, different cumulative display. So we can see that during initiation, the rotors overall showed a shortening of cycle lengths before the rotor and after the rotor. This is the red arrow. And then there was a continuous acceleration from initiation to early stabilization. But once you have early stabilization, there was no difference between before the rotor and after the rotor. The role of the rotors is accentuated or demonstrated by the fact that when we did the same analysis for a singularity point that did not form rotors, there was no effect before the singularity point after the singularity point. So the cycle length did not change in the presence of singularity points that did not form rotors. So we ask the following question. If rotor accelerate the atria, and those that are driving the sustenance of the AF, then what distinguishes rotors that accelerate the atria and that could serve for targeted therapy? Well, the short answer before I show the data is that we don't have full answer for that. But here's what we try to do. First, we try to look whether they distribute in certain regions, those that accelerate. These are before during the initiation, we see there is a slight more rotors actually in the right atrium, although the tachypacing was in the left. But they were both in the right and in the left. While in early stabilization, there seems to be equal number in between the left and the right. So there's no significant difference. So we try to look at the drift. Maybe the drift is different. Here we have a trajectory of the rotor, and we look at the vector that connects the beginning and the end of the drift, and we look at the magnitude and the velocity along that drift. And what we notice is that indeed the magnitude of the drift is larger for those that accelerate compared to those that decelerate, both at initiation, but not when you have early stabilization. We also see the velocity for those that accelerate is larger than those that do not accelerate the atria. But during early stabilization, that's where you electrophysiologists in the clinic see the patients. There is no difference between those that accelerate and those that do not accelerate. So probably we have no way to distinguish between them. So in conclusion, what I've shown you that in a sheep model of cholinergic atrial fibrillation onset progression, the frequency and phase domains analysis provide insight into spatiotemporal excitation patterns driving AF, mostly rotors, as I showed you. We've seen serial accelerating rotors that cause net atrial cycle length decrease and drive the dominant frequency increase during AF onset. And biatrial panoramic is critical. Biatrial panoramic mapping reveals that rotors that accelerate atrial fibrillation are distributed almost equally across the two atria. But rotors that accelerate the two atria are drifting more and at a faster velocity than rotors that do not accelerate. Thank you very much for your attention. And I'm sorry I forgot the most important slide of acknowledging the many people and support on this study. Thank you again.
Okay, now it's my task to announce the next speaker, Dr. Natasja de Groot from the Erasmus Medical Center in Rotterdam. She's a professor in translational electrophysiology and a Delta medical professor. And she'll talk about insights from high-res, high-density mapping of atrial fibrillation in humans.
Thank you very much, Pascal. Good morning, ladies and gentlemen. It's also a pleasure to present here on my favorite topic, and that is high-resolution, high-density mapping of atrial fibrillation. And in my presentation, I will summarize what we have over the past years learned from this technology. So it all started in 1997 with the group of Maurits Alessi, and he developed a spoon-shaped electrode, which you see here, which contained 265 electrodes with an interelectrode distance of 2.25 millimeter and a diameter of 0.3 millimeter. This electrode was used to map patients who at that time underwent cardiac surgery for the Wolff-Parkinson-White syndrome, and in these patients atrial fibrillation was induced. And he showed for the first time in humans that during atrial fibrillation there was so-called random reentry, which you see on the left part of the slide, where you can see that the wavefront comes from the right to the left, encounters a line of conduction block, but then it's re-excited by an activation wavefront which propagates from the opposite direction. And that's what they called random reentry. At the other part, you have the leading circle reentry, where you have a wavefront propagating around an area of conduction block. A few years later, we used the same technology to perform mapping procedures in patients with long-standing persistent atrial fibrillation who underwent cardiac surgery for valvular heart disease and/or coronary artery disease. And we then realized at that time that people were talking about the substrate of atrial fibrillation, but it was not yet well defined what substrate actually means. Because we can quantify all kinds of parameters, which we do by fractionation mapping, by voltage mapping. But I think the first step is to simply quantify parameters measured during atrial fibrillation and also to do that during sinus rhythm and compare those values. So this was the first step in, if you have two different types of atrial fibrillation, can you actually distinguish these two types of AF by quantifying parameters? Well, one of the first parameters, of course, the number of fibrillation waves, which is quite simple, and indeed we showed that if you have long-standing persistent atrial fibrillation, you have more fibrillation waves, but also a smaller number of fibrillation waves. In the study, we also demonstrated that patients with persistent atrial fibrillation have so-called more focal fibrillation waves. It was even 4 times higher in the patients with persistent AF. And based on these observations, we introduced the so-called double layer hypothesis. And we called it double because at that time we could only measure at the endo and epicardium. And we said, well, if atrial fibrillation becomes more persistent, you develop electrical asynchrony from the layers. And because you have electrical asynchrony, fibrillation waves can propagate from one layer to the other, thereby acting as an ectopic beat at the other side. And this will perpetuate atrial fibrillation. So we demonstrated that in a series of patients that indeed when atrial fibrillation became more persistent, you have a higher degree of endo-epicardial asynchrony. But it became also more difficult because we started with simultaneous endo and epicardial mapping of sinus rhythm, and this is what you expect. You see an endo and epicardial activation map, you have the corresponding electrograms, and they look alike. And that's logical because the atrial wall is only a few millimeters thick. And here you see an example. So this is an asynchronous activation during AF. But on the other part, you see, hopefully, if I press again, oh, sorry. Well, it skipped the movie. But at the other part, you saw that there was a part of asynchronous activation, even during sinus rhythm. And that is what we also see here. On the left part of the slide, you see a transmural conduction block where the conduction line is across the endo and epicardium, and you see a similar pattern of activation. But in case of an asymmetrical conduction block, you see that the AP and endocardial is completely different activated, and that gives the electrical synchrony, and that is only during sinus rhythm. So then we start to analyze the endo and epicardial asynchrony, and here you see some typical examples. You have one example, we have synchronous activation during AF, and you also see that the AP and endocardial electrograms, if you make an overlay, are quite comparable, but in the patients with more the persistent types of AF, you have asynchronous activation, where if you overlay the electrograms, you can see that they're quite different. But also, most interestingly, that not there is only an electrical asynchrony, but even in the small, thin layers ASIA, you have electrical fibrillation waves propagating in opposite directions. And I think that's still quite remarkable. So, based on these observations, we realized that the substrate of atrial fibrillation was far from understood. So we initiated the QUASAR project, which is the quest for the arrhythmogenic substrate of AF, and that is what you see here. In all the patients who undergo cardiac surgery in our hospital, whether you are in sinus rhythm or whether you have AF or whatever underlying disease you have, if a patient wants to participate, it undergoes complete epicardial mapping according to this scheme. So we have here for an electrode array, which contains 192 electrodes. And we shifted from the right atrium, because we shifted across the left atrium, the pulmonary vein area, and also across Bachmann's bundle. And here is what you see. This is just a few examples. So what we want to do is to compare all variables between the different types of atrial fibrillation. And just to show it very quickly to you, we gave them the colors of the Dutch flag. So if you are on average, it's white. If you are higher and you go in a parameter to electropathology, it's red. And if you are below the average value, but it means no electropathology, it becomes blue. And so this is a patient with no history of AF. You can see the red boxes for the specific parameters. And then we start to compare it with patients with persistent AF. And as you can see, this is the several parameters are becoming more in red. But this is, of course, a very coarse approach, and we are now refining this methodology. But for this presentation, because I noticed that Dr. Berenveld was presenting before me, and I thought, well, it would be nice because they are presenting on AF, on the rotors of AF, let's try to find in my population if I can find a driver using criteria which are used in the cath lab. So simply the extracellular potentials, and they say that if you have a short atrial fibrillation cycle length and a regular activation, it's associated with drivers of atrial fibrillation. But if you take a critical look at the literature in the clinical studies, the relation between these parameters and the underlying patterns of activation is incompletely understood. So I asked one of my PhD students to go into this, and she had 71 patients with different types of atrial fibrillation, so paroxysmal persistent and long-standing persistent, who all underwent cardiac surgery. And in those 71 patients, we performed epicardial mapping of the entire epicardial surface. So we ended up with quite a large number of recording sites, namely 93,000. So that's on average like 1,300 sites per patient. We had to exclude above 15% of the recording sites because we had some loss of contact. At each site, 10 seconds of AF was recorded. The median cycle length was 106, and we found a median standard deviation of all fibrillation cycles of 35. So we started to analyze the patterns of activation. We classified them as focal activation, smooth propagation, propagation with conduction block, collision of fibrillation waves, and remnant activity. We didn't know how to call it, but we frequently observe that in patients with persistent atrial fibrillation, you have very tiny fibrillation wave covering only a few electrodes. So we provided them with definitions, but because my time is going quite fast, I will skip this. And we related it with the electrogram morphology. So we classified them as single potentials, double potentials, and fractionated potentials, and we measured also voltages. And this is what you see here. You see 4 typical examples where we measured the median cycle length at each electrode, and we went in search for the shortest AF cycle length. And as you can see in red in each patient, there are multiple areas with short atrial fibrillation cycle lengths. Then we started to localize them and to measure, and we measured that the shortest cycle length in each patient ranged between 65 and 183 milliseconds. And if we take a look at the site where these shortest cycle lengths were recorded, they were mainly found, and not surprising, the pulmonary vein area and the left atrioventricular groove. Interestingly is that the sites of these electrodes range between 1 and 3 electrodes. The screens here are flashing on and off, but doesn't matter, I will take a look at the other screen. And meanwhile, we also wanted to know whether the patterns of activation were regular, so we also identified the areas with the smallest standard deviation. And the smallest standard deviation in each patient ranged from 1 to 15 milliseconds. And here you see that these sites were located mainly at the left atrioventricular groove, but also frequently on the right atrial appendage. So then we combined them, but because we wanted to know if you have the shortest median atrial fibrillation cycle length, is that also the most regular atrial fibrillation cycle length? Well, these are the 4 examples, and they are the same patients, so each median cycle length corresponds to the standard deviation. And just by comparing the colors, so the red color should be the same in each picture, you can already see that there is a mismatch between the shortest atrial fibrillation cycle length and the most regular activated areas. Are the short median cycle length indicative of focal patterns of activation? And this is what you see here in this table. So for all the different patterns of activation, we analyzed the median atrial fibrillation cycle length and the standard deviation, and we found that the focal patterns of activation did not occur more frequently either at sites with the minimal atrial fibrillation cycle length or the most regular activated site when you compare it to the remainder of the atria. And the largest proportion of focal atrial fibrillation waves was also not located at these sites. But then it raises the question, if you find during mapping, so simple mapping as we do it at the cath lab with electrograms, what do we find at the shortest median atrial fibrillation cycle lengths? How are they different from the remaining of the atrial tissue? And we found they have more areas of conduction block, they have more areas of so-called remnant activity, so the very tiny fibrillation wave, and we often found more long double potentials, which of course indicated areas of conduction block. We saw a decrease at this site in the propagation wave, or smoothly propagating wave, fibrillation wave, without any areas of conduction block, a decrease in the number of single potentials, and also in the sharp double potentials. And then what are the regular activated sites if you compare them to the remainder of the ATIA? There we saw that there was an increase in the smooth propagating fibrillation wave, there were more single potentials, there was a decrease in the areas of conduction block and remnant activity, and also a decrease in the number of long double potentials and fractionated potentials, which are indicative or areas of conduction abnormalities. Now, the last step is then to identify if we could find repetitive focal patterns of activation. So after we identified the areas, we counted that there were like 90% on average proportion of focal fibrillation waves. And here you see the distribution of these focal fibrillation waves, that there were surprisingly mainly found in the right atrium and the left atrioventricular groove, and the smallest amount was found at the pulmonary vein area. And then we decided to determine how often they occur consecutively, because that might be indicative of a driver. And we found here that the range of series of consecutive fibrillation waves ranged between 2 and 28%. I'm sorry, 28, not percent, but the number. And here you see where they were located, and then they turned out again to be mainly predominant in the right atrium. Also, there was no difference in the atriofibrillation cycle length of these consecutive focal fibrillation waves. And for some reason, we found more short double potentials, but I don't know how to put that yet. So based on this clinical study, we could say that the fastest or the most regular activated areas did not correspond to the repetitive occurrence of focal patterns of activation during atriofibrillation. So the findings of our study does not support the concept of detecting drivers by identifying the sites of the fastest or the most regular activative recording sites. Thank you for your attention.
Thank you, Natasha. Our next speaker, that will be a pre-recorded session by Natalia Trajanova, and she herself is in transit from the airport, but probably will be joining us at least through audio communication later.
I'm really delighted to be here, and I am very grateful to Dr. Elad Anter as well as the rest of the organizing committee for inviting me here to present our research. I really wanted to be in person at this meeting. I love it. It's one of my favorite meetings, and I am suffering from FOMO, fear of missing out, because I know you guys are going to have a lot of fun at this meeting. So today, I would like to share some results from our research that provide potential insights how we can improve substrate characterization for VT ablation. For patients with ischemic cardiomyopathy, there are a number of ablation strategies in use: core isolation, substrate homogenization, identifying electrophysiological abnormalities and ablating them, as well as ablating image-based channels. Despite this wider use, there is a need for further improvement in VT ablation, but that improvement would require further advancements in our understanding of the VT substrate, which we are hoping to provide. So there have been new perspectives in VT pathophysiology in the last 5 years, let's say. For instance, a very important study by Rod Tung that demonstrated this transmurality of the propagating wavefront with different characteristics and exit size isthmus on the epi and endocardium, as shown here, as well as the insights provided from the work of Dr. Elad Anter, demonstrated that there is an important functional aspect to the VT circuit. In terms of the functional aspects of the substrate, these functional aspects actually really affect how we do the assessment of the substrate. For instance, here in this study, this is the physio-VT clinical trial, Dr. Anter and his colleagues demonstrated that the pacing site, the location of the pacing site, affected the regions of slow conduction, their location, as well as the magnitude of slowing, and different wavefront directionality was able to uncover new locations of conduction slowing. Furthermore, in the study of Srinivasan and colleagues in 2020 demonstrated that an extra stimulus giving at some time after the initial pacing stimulus unmasks better with higher accuracy the locations of critical locations of the VT circuit. It's much better than just sinus rhythm evaluation. So how do we integrate all this new information in some unified framework to study the VT substrate? And for those who are familiar with my work, you guys know that we do what we call a virtual heart approach, or also called heart digital twin, where we reconstruct patients' hearts, typically from imaging modalities, and also we incorporate some data for personalization of the electrophysiological properties. We construct these patient-specific models. We do arrhythmia or pacing simulations, and then we are able to provide with them mechanistic insights as well as suggestions for novel therapies or clinical support. So I'm going to be using here a very similar approach, our virtual heart approach. So the goal of this study was to investigate how multiple wavefront pacing, decremental pacing, and the combination of both improves identification of patient-specific disease-induced remodeling and critical VT sites. We conduct these analyses in this personalized heart model, so the transmurality, the three-dimensionality of the VT is naturally accounted for. And then we provide assessment of how best to apply functional mapping strategies to optimize identification of VT ablation targets. So our approach consisted, as I said, of creating these patient-specific hearts. We actually, in this study, used 48 models of 48 patients and reconstructed them from imaging modalities. Here you can see the distribution of the zone of infarct. And then we did substrate mapping by wavefront propagation from different pacing sites. Given that this is a simulation study, we were able to stimulate pace from many locations. Specifically, we paced from 100 different locations. The rationale for doing that is it's an assessment that cannot be done in the clinic. We have 50 pacing sites on the epicardium, 50 on the endocardium, and we were able to explore directionality of the wavefronts as well as location of the pacing site with respect to the infarct. We also explored decremental pacing. You can see here, in addition to just S1 protocol, we had S1 plus S2 at 360 milliseconds and another protocol where the S2 was a little bit shorter. We executed over 14,500 pacing simulations just from these many pacing locations and for these different protocols. Once we conducted that, we actually used the simulations to recreate electroanatomical maps. We did calculate from the simulated propagations, we calculated electrograms on the epi and endocardial surface. And then we use these electroanatomical maps to extract features from these electroanatomical maps and create maps of features. So here you can see this is a map of voltage amplitude, a map of conduction velocity distribution, a map of isochronal crowding, a map of fractionation index, electrogram duration map, and frequency power map. So we created these for each pacing site. We created 6 maps from the electroanatomical mapping features. When this was done, we conducted comparison of the different features, electroanatomical mapping feature maps, with the location of the infarct as it is on clinical imaging, as well as the location of the critical VT site, the critical circuit sites, as calculated from the model. So this is a separate calculation where we calculated the VT and defined its morphology using these isochrones. Doing all that, then we completed the analysis doing logistic regression models to compare whether the maps of electroanatomical features correlated with the infarct remodeling and also with critical VT sites as well as with itself. For instance, whether a map acquired by pacing from one side was similar to a map acquired from another pacing site. So we'll start with that actually and first demonstrate whether there was a concordance between electroanatomic feature maps for different S1 wavefront directions. So that was the first result in this study. So here you can see the concordance shown from 0 to 1 of all these 6 electroanatomical mapping features. And on the horizontal axis is the extent of the infarct. It's the remodeling burden in these hearts. So you can see here that for hearts that have small infarcts, the concordance between 2 different electroanatomical maps from 2 different pacing sites was not great. For none of the features, it was particularly good. But as the size of the infarct increased as a percent of total volume of the ventricle, as the size increased, you can see we get better concordance in all of the features, particularly actually in conduction velocity and isochronal crowding. So for bigger infarcts, we get better concordance between maps acquired from 2 different pacing sites. So to answer this question, you can see here it very much, this concordance depends on the extent of infarct remodeling. For small infarcts, it didn't work very well, but the substrate characterization was similar for different wavefront direction if the zone of infarct was large. Here are 2 examples. I am showing one heart that was paced from 2 different locations, one left ventricle paced from 2 different locations, and another left ventricle also paced from 2 different locations. This one had a larger infarct than this heart. So here you can see a map of voltage amplitude. The 2 maps are fairly similar for the 2 pacing locations. Similarly for conduction velocity, isochronal crowding, there was a concordance, fractionation index, and so forth. But for the heart with a small infarct, you can see there was basically no concordance in the voltage amplitude, not much in conduction velocity, and so forth in the other features. So exactly, this is an illustration that for small hearts, for small infarcts, that the concordance was not really good. So then the question that we asked next was, does detection of the infarct by these electroanatomical features depend on the distance of the pacing site to the infarct? And first, we looked at hearts that have bigger infarcts, and you can see in these hearts, there was basically no dependence on the location of the pacing site to the infarct. This is area under the receiver operating characteristic, F1 score, sensitivity, specificity, pretty much does not depend on the distance to the pacing site. In hearts with smaller infarcts, actually the finding is the same, it does not depend. But looking at these 2 different results, you can see that how much worse are the results for F1 score and how unbalanced sensitivity and specificity are in predicting the location of the infarct in hearts with smaller infarcts using these electroanatomical features. So the electroanatomical features from a single pacing site detect the infarct poorly in hearts with smaller infarcts. So is that detection improved by multiple wavefronts? The answer is yes. You can see there is immediate improvement here, and you don't have to do too many pacing sites to see improvement. After that, the curves level off. However, this improvement was better in hearts with lesser infarct. So the multiple directionality improved detection for all infarct sizes, but the incremental improvement was larger for smaller infarcts. And this is an example here. Larger infarct, smaller infarct. Gray represents an area where there is missed prediction. You can see as the number of pacing sites increases, the prediction gets better, but it's so much worse in hearts with small infarcts. Then is detection improved by decremental pacing? Now we are adding a stimulus, we are adding S2 at different timing, and even here we added 2 S2s. So let's see what comes out of this. What you will see here is for the decremental pacing, these 4 histograms are for hearts with large remodeling, large infarct, and these are for small. With small infarcts. And you can see the number of pacing sites, the multiple wavefront propagation, increased a little bit the accuracy of prediction for larger infarcts, but improved it pretty significantly for hearts with a larger infarct. So as we gave S2 stimuli here, we got F1 score that was closer to 1 for all these different features. So the combination of multiple wave propagation and decremental pacing improved the detection of the infarct zone, particularly for smaller infarcts. So is the detection of the inner VT circuit improved by that as well? Here is how we separated the inner and outer VT circuit very much by the methodology of Rod-Tung, and we found that multiple wavefront propagation did not really, only very marginally improved identification of the critical VT circuit. You can see here the increase of pacing sites. However, when we added the decremental pacing, it really immediately improved. The features became much more predictive of the inner circuit. So combining these two greatly improved the critical VT detection sites. So the take-home message here is that the VT substrate with smaller infarct exhibits more variability to functional electroanatomical characterization than hearts with bigger infarcts. Electroanatomical mapping with multiple wavefronts is more advantageous in smaller infarcts. Decremental pacing is synergistically can combine with multiple wavefront pacing to improve detection of the remodeled substrate, and decremental pacing from multiple locations greatly improves critical VT identification. This is the paper if you all are interested, and I end with these acknowledgements. Thank you very much.
Our last speaker of this session is Olaf Dossel. He's professor and head of the Institute of Biomedical Engineering at Karlsruhe Institute of Technology at Karlsruhe, Germany. And Olaf's talk is signal recordings, ideas to improve catheter designs.
Thank you very much to the organizers for inviting me. And one remark in the beginning, I suggested to you, Elat, 2 other topics which were related to publications we had this year, but you have chosen this one. And this is very similar to the one that I had last year, so don't be surprised if few of the slides are the same as compared to last year. But that doesn't mean that I'm not happy about this topic. I like it very much. So where is it here? The forward is this one? Yes. So this is the outline. Why and how do we simulate electrograms, varying electrode size, thickness, distance, and there you will see a lot of things that Pascal has already told you, and so I can be very brief. Then I move to arrays and matrices of electrodes. What do we really need? And in the end, I conclude with what does all this mean for the optimal multi-electrode catheter. I will concentrate on atrial signals, and I will concentrate on contact mapping in this talk. And this, I think you've seen before. I like it, and it's very much similar to something that Pascal has shown. It's a computer model. The atrial tissue consists out of, 100,000 individual cells, Courtemanche cell model, and it's the simulation of a propagation of a wavefront. You see the unipolar signals and the bipolar signal, and if I stop here or make a sign here, you see that the moment when the wavefront is passing by the distal electrode is where we have the steepest negative slope in the unipolar signal, and it does not precisely coincide with the maximum of the bipolar signal. That's, but it comes quite near. This is from a study where we had, I think, 100,000 electrograms and make a cluster analysis. So real clinical data, not simulated signal, and we see this unipolar signal and the bipolar signals. We see the same shape, that morphology that we have just seen in our simulations. We observe a typical RS shape in all unipolar signals and a large variability in bipolar signals. And if the wave comes from underneath, the R wave will be missing. And if the wave is approaching a block or goes down into the tissue, the S wave is missing. So we can see a lot of interesting things in the unipolar electrograms. That has been pointed out already. And this is now a very simple model of fibrotic tissue. In the upper line, you see the transmembrane voltages, and in the lower lines, you see the extracellular potentials. It's as I admit, it's a very simplified model of fibrotic tissue. We have, meanwhile, very extended models, but the basic message that I want to tell you is already seen in this simple simulations. The effective conduction velocity drops significantly, and the signals become more fragmented with increasing degree of fibrosis. I constantly use the term effective conduction velocity because we had this discussion in the last session that there is a real conduction velocity through the viable tissue and an effective for the whole tissue, and I always refer here to the effective conduction velocity. And these are the signals, the simulated signals that belong to this fibrotic tissue with 30% and 40% fibrosis. And what do we see? The signals become more complex and fractionated with increasing degree of fibrosis, and the signals, the activity duration becomes longer with increasing degree. And what I think is also worth to mention is that the similarity of neighboring signals becomes smaller with increasing degree of fibrosis. So the different colors here are neighboring electrodes. If you only go 0.25 millimeter away from the first electrode, the signal is still very similar. But if you go a few millimeters away, the signals have nearly no similarity at all. And my question is then, what happens to the bipolar and omnipolar signals? If you remember all the interesting things that Pascal said, that's all true for healthy tissue. For example, if a wavefront is approaching both electrodes simultaneously, you get no signal in the bipolar signal. All true for healthy tissue, not true for fibrotic tissue, because if you subtract two signals that are not similar, they cannot cancel each other. Varying electrode size, thickness, and distance. This is something that I've shown to you last year already, and so I will be very brief. So we changed the distance, center-to-center distance of bipolar electrodes. Both are touching the endocardium, the bipolar and the unipolar distal signal, and you can see that the signal increases with in the bipolar signal with the distance, but we have also seen if you increase now further to 5, 6, 7 millimeters, you will get two unipolar signals with inverted sign. And so larger electrode distance leads to larger bipolar voltages, as long as you are not too far away, and reduced field cancellation. Pascal, I think, also pointed that out. This is maybe more interesting now, variation of the cubic electrode size. So we increase the metal above the electrodes, and you see that if you increase the electrode size, the signal goes down. This is maybe not intuitive to you, but from numerical field calculation, it's absolutely clear, because this metal object will distort the field, and this will lead to exactly this effect. So small is beautiful. Smaller electrode size leads to larger voltage. In particular, the electrode height is important. So if you see the picture with the tissue and the height, and if you increase the height, the signal goes down. So what is the message? Thinner electrodes lead to larger voltages. And this is, of course, also known to everybody. If the distance to the endocardium becomes larger and larger, the signal goes down. And I show this to show you that even with the distance of 1 millimeter, you already get a significant decrease in voltage. Signal drops significantly. Bipolar signals drop faster than unipolar. Now we have learned that maybe unipolar signals are the better choice. I admit fully, I fully agree, if you want to make activation time images, then it's the better choice. If you want to make voltage maps to decide where are low voltage areas, I refer to this study that has been published already 2 years ago from our group with many patients. So a real clinical study. On the left side, the anterior view, on the right side, the posterior view, and in the upper, the bipolar voltage map, then the unipolar voltage map. And if you then agree on the threshold and say, I classify 0.5 millimeter to be low voltage, and then compare the two maps, they overlap completely. So this is the reason is that if you go to extremely low voltages, then you go to fibrotic tissue, and then the bipolar signals do not cancel anymore. And so you can easily use the bipolar voltages. But if you want to agree worldwide on a threshold, then you have to decide whether you look to the unipolar or the bipolar, and you have even to agree on some adjustment referring to the catheter that you use, referring to the metal that is in the catheter, and so on. So you have to adjust the threshold for multi-center studies, but otherwise you can use unipolar or other voltage maps. Now, this is about the transmural and epicardial fibrosis, and so we made some simulations where we either had a transmural fibrosis in the lower left corner or just epicardial fibrosis with the electrode on the endocardial side. And here you see the picture shows on the x-axis the diameter and on the y-axis the voltage. You see that with the fibrotic tissue, the yellow curve, you really get a very low signal. But if you only have an epicardial fibrosis and an endocardial electrode, you hardly don't see any difference. And it doesn't really change if you increase the electrode. You heard the rumor that you can see to the other side if you increase the size of the electrode. It helps a little bit, but not really. So this means we are as a little bit blind, let's say, about what happens on the other side of the electrode. So this reminds me of a publication that we have here in October 2022 from Natasha and Matthijs, I've heard, is also in the room, the first author. And the main message was different from what I want to tell you. The main message was unipolar and omnipolar low voltage areas are frequently located exclusively at either the endo or the endocardium, epicardium, so mismatch. My comment is endocardial measurements cannot really see what's going on the epicardial side. This study, I think, is another argument that is convincing. If you then say, okay, then we switch to lead gadolinium enhancement MRI, I say it does not really help, because I always claim already for 5 years, I think, that lead enhancement MRI is not quantitative due to the partial volume effects. You see the voxel size of the MRI and the size of the tissue and the size of the fibrosis. And you can read something from that pictures. They have some worth and some value, but they are not quantitative. Arrays and matrices. I have to be fast. I see again, I have time problem. Arrays and matrices, that is what we have. I should have added the the pentarray, the octarray. We'll do it next time. It is a major step, I would say. You see the size, the 20 millimeter overall size, and the distance, 2 millimeter, 3 millimeter, 4 millimeter. I restrict myself on contact mapping, and I also do not comment so much on this constellation or firm map, because I think the distance of up to 20 millimeter between electrodes is much too large. But I like the Peter Spector's electrode sandwich, I call it, and it's a stacked, and I think, Sonny, you were referring to that without telling that Peter is he in the room? They are trying to sell that, okay. So how can you visualize the spread of a wavefront? We can either do sequential high resolution mapping of repeating patterns, or if they are not repeating, we can do simultaneous acquisition in a large area with a coarse resolution, or simultaneous acquisition with high resolution, but with a small coverage. And we did a study here sponsored by Boston Scientific with over 300,000 electrograms. And here you see the bipolar voltage, activity duration, entropy, which is a measure of fractionation. And on the x-axis, we see the distance to some important point. And in this case, it was a microchannel. And you see that in a distance of up to 6-7 millimeters, the signals are changed. And if you now imagine that we only measure every 5 millimeter, we would not capture the whole story that is in this picture. So 2 millimeter spatial resolution would be really nice. And I skip this one because of time. And I refer here again to another picture that I think also comes originally from Matthijs and which we have seen a few minutes ago. If you look to the conduction velocity map, how can you pick up such a beautiful map with some matrices with a 4 or 5 millimeter distance? I would say impossible. We cannot see all these details, and these are real human data. It's the reality that we have to face, and my message is it would be nice to have this resolution and area in a clinical environment. We just heard a lot about conduction velocity from Natalia, and we picked that idea was already clear from the beginning that conduction velocity is anisotropic. And here is a study where we stimulated with a lasso catheter from several directions. In the picture, you see just 3. And then we can really pick up the anisotropy. And in the lower corners, you see the conduction velocity and the voltage as a restitution curve. And so we also had this S1, S2 stimulus, and then we can really see how, with which interstimulus interval, the conduction velocity really goes down to 0, and the voltage really becomes small. And, whoops. Well, so maybe I move on with this one. We think that we can, with a dense matrix, distinguish between atrial near fields and far fields. We have also heard a lot about that from Sonny here. We have an algorithm that can distinguish atrial near fields from atrial far fields, provided we have a dense electrode matrix. We can also use machine learning with a dense matrix, and we are convinced that we can, on the long run, distinguish between transmural fibrosis and fibrosis that is only on one side. And we think it will be also important in the future to solve the inverse problem for the electrodes that are touching the endocardium. We can still solve the inverse problem and go down really to the sources that create the signals, the dipole layer, and by that get a better spatial resolution. What does all this mean for the optimal catheter? On one side, we can make measurements in sinus rhythm and atrial flutter or in pace rhythm. And there we can always shift the grid and get a very high resolution with a coarse catheter. And we can also get a good coverage by moving. So high spatial resolution and large area coverage is nice to have, but we can go and with the catheters that we have today. But if we want to make measurements during AFib, then we can do voltage maps, activity duration, all with the systems that we have. As soon as we want to go to spatio-temporal analysis, it's only possible with small spacing and large coverage. And that refers to conduction velocity maps, phase maps, cycle length coverage, similarity of signals. There we need the real spatio-temporal coordination. And what does this all mean? I'm dreaming of a flexible electrode grid. Why? Because we have to touch the endocardium with 2 millimeter spacing that is covering an area of at least 20 millimeter diameter with electrodes of 0.2 millimeter size. Small is beautiful, and very thin gives you a high voltage. Thank you very much.
Olaf, you want to join us?
Okay.
Yep.
I'm here.
We are now open for discussion. I will say that we are running a little bit late. So we're going to go on until you tell us to go ahead and stop. Andre.
I do have a short question or a remark. The intrinsic deflection was defined at the beginning of the last century, I think, by Thomas Lewis, when people did not know what an action potential would be. And then much later, there are, I think, 3 studies, one by the Amsterdam group, Joram made a study, and Madison Spach, showing that the fastest deflection in conductive tissue does not necessarily correspond to the maximal of sodium current flow. So there is a small inaccuracy there. If we do in cell cultures, produce heterogeneous coupling, and some cells are separated from the other, we can record within a few micrometers local delays of 80 milliseconds, certainly. Could show you many recordings. In addition,
Delay from the actual activation
well,
time to before you see
between
the...
activation of cells, one cell to the other. And Joram showed in his simulations very long delays within coupling. We also always see multiple humps in a single cell of membrane. This is membrane potential, and it's accurate because we can map with an accuracy of 6 micrometers. So I have a little bit of provocative question. Can we even measure local activity? If within a short distance you have scattered signals, and they will report these membrane potential changes in the extracellular space as multi-spike signals within a very, very short distance. So what is local activity? I understand that you need local activation. I would like to know the same, but when you have very large heterogeneity, it may be actually difficult to define it. I don't know, maybe also what Ed thinks, he did also simulate this. I know it's provocative, but...
It is what it is.
I think even at the cellular level, if you're talking, when does a cell activate if you have different parts of the cell activating at the same time?
Yeah.
So it's a basic definition.
Omer, good.
Thank you, Andrea, for the comment. I think a guidance could be probably taken by the fact that the space constant in tissue and the width of the front is of the order of 1 millimeter. Obviously, it's a guidance. It's not. It doesn't say that there is no variability with less than that space scale.
I mean, Stefan Rohr in Bern has tried to use voltage ties marking ion currents, not voltage. And he has managed to visualize inflow of calcium. It's all very high resolution, but it may answer, it's for me a difficult situation. Because you may look for an absolute index of local activation, that would be difficult to define at the cellular level.
But well, from my point of view, it's a different view to the story, whether you are interested in basic research and understanding in a scientific lab everything about the processes, which is very important, and that's maybe what you're talking about. And many of us were talking about the clinical situation. The patient is on the table, you have to do something, what is reasonable, what do you need? And that's a different point of view. And we sometimes use terms of activation time and conduction velocity that are used, the same words used in different contexts. And that makes the discussion a bit difficult.
That does not solve the problem for me. I don't know, Pascal.
Yes, no, I hear all these smart remarks, and I guess what Olaf says, that's probably true. I think that we, of course, also have to work with a model of reality, and that allows us to define the moment of local activation. But I fully agree that when you measure things at your level, then that probably is an incorrect estimation. But what we have and what we need.
But Sunny and Pascal, may I ask you? If we are talking about measuring the local activation time, and we now have mapping systems where the sampling rate is rising, so you have your steepest negative deflection, and there are several samples within. So what is the correct way to calculate it, going from your positive to your most negative point and calculate there the difference, or do we calculate the steepness of every segment and then take the steepest deflection? Because there's also a difference in that, and I think there's no literature on that, what you should do.
I am going to defer that to Pascal.
Yeah, thank you.
Sorry, Pascal.
Yes, can you repeat the question again?
So there are two ways. If you think about, okay, we were talking here about determined local activation time. You calculate your steepest negative deflection. But if you take a look, if you have an electrogram, you have a positive deflection, the steepest negative deflection, and then the negative point. So you can calculate the steepest deflection as the difference between the minus and the plus, or between all the different segments, because the sampling rate is going up. So you have different deflections. And what are we taking now? What is the correct way to do it? Because I'm not aware of literature on the topic.
In your slides, you showed continuous conduction. And there the situation is clear. But then when you want to determine it, you speak of discontinuous conduction. So where is your model of discontinuous conduction showing the theoretical curve?
I think in general, you would select, of course, if the signal is complex, then it's always difficult. But in general, you should, I think, select the most negative value. So that's what I do.
So there's 10 samples in your deflection, you just take that sample. Because perhaps we should look into that, because I'm not aware of any data.
No, that's true. Yeah, and of course you're working.
Be a practical matter for what's doable.
Yeah,
exactly. And by definition, you're working in a discretized system. So again, also depends on your sampling frequency and what you can measure.
Or could it be that plus minus 1 millisecond is clinically irrelevant?
it is clinical. Well, it might be, but if you have your signal becomes broader, then of course you might have some inaccuracies. And if we're talking about determining conduction velocity, conduction block, a few errors add up in your final calculations.
Yeah, and that's actually, I want to follow question. And first of all, congratulate all speakers. It's a great session specifically. And Olaf, when you mentioned regarding actually your dream of electrode and defining that you're using this electrode would like to define activation and conduction velocity, because it echoes all this discussion. Conduction velocity, even in Atria, it's three-dimensional. And that's like what we see in optical mapping, what Big One just present, showing this three-dimensionality everywhere. So how you even can measure conduction velocity? Like using any type of electrode when in human, especially human left atria, up to 1 millimeter fibrosis and the fibrosis. And then already you have electrical activation going through multi-bundle insulated. So but electrodes, what you dream, the touching endo
Yeah.
So how this would be interpreted?
Yes, the interpretation.
Because all the signal,
yes.
what we're talking about fractionation, where's this like
Understand.
golden standard validation?
Yeah,
How
yeah.
it's going to be?
Yeah, I understand, but well, we have, I think, also a couple of publications, and Caroline Roney is, I think, the leading lady in the world who knows which algorithm you should use to calculate the activation time, calculate the conduction velocity from a couple of activation times. And all these methods have their errors. And for the interpretation, I would say we can only see what happens in the first millimeter layer. And what's deeper in the tissue, we don't know.
Yeah,
We
I
just
agree
don't
with
know.
that.
We are
I agree
blind
with
for
the
that.
electrodes.
Or you use epicardial electrodes.
But if you do simultaneous endo-epicardial mapping, sometimes we have normal conduction on the endocardium and a clear epicardial conduction block. You can see that on the endocardium as a far field. So
As a
we
far
are,
field.
as a
Yes.
far field. So you
And
can see what is happening transmurally.
Yes, so that's why I said, I mean, in the last sentence I said with some kind of machine learning, you have to identify the far field from other fields. And I think with machine learning, we can solve the problem in the near future and see how the signals in the spatiotemporal matrix look like, and then have a good guess of what's going on the other side. But basically, I wouldn't say we are blind for what's going on the epicardial side, but it's hard to discover.
Well, the problem with the far field is that you don't know the direction. So it could be epicardial or it could be over here. Yeah. I mean,
Because it also depends from fiber orientation. When we see in the human, fiber orientation would actually change the far field because space constant along the fibers anisotropy, then actually perpendicular would be different. And when we go into human remodel arithmogenic, when it's more insulated, it also changes significantly the space constant.
So I mean, I think that plays into what Natalia Tryanova was doing with the multiple pacing sites. You're going to be exciting at all the different directions, and then something that's more structural will come out as opposed to something that's just functional based on stimulation site.
Yes, that's a very important issue. I think I made clear, but we published that, and you can even download the sequence and the algorithms from the internet that you stimulate on a lasso several times from several sites, and then you even know the fiber direction in the upper layer, because that's the direction where the conduction velocity is the highest. So you don't get the conduction velocity of just that pattern that randomly passes by, but you get the conduction velocity for all directions. And together with the S1, you get also the restitution curve, which is the same important, because during AFib you have a very fast depolarization, and then all these values, the conduction velocity, and they are all changing. Yeah.
Yeah, but you can't really measure conduction velocity during AFib. So you have to actually use pacing, because conduction velocity measurement during AFib, when you have very complexity going between AP and endo, it's not literally possible to do properly. Yeah. We're talking about conduction velocity measurement during pacing, sinus rhythm, right? Oh.
Yeah, I did not understand 100%, but what you mean. But if you say again that we are kind of blind on what is going on the other side.
No, I'm only mentioning about conduction velocity measurement during atrial fibrillation. So it's a question because it's going from different directions. It's conduction velocity measurement should be done during pacing, and during pacing from different locations together.
Yes, what I meant was you bring the patient into a sinus rhythm. We started a couple of years ago already with the coronary sinus, where you can stimulate in 1-2 and in 9-10. And then you already get 2 directions, which helps a lot. Yeah. But the best way is this new technique that I proposed.
Thank you all. Thank you.
Yeah.
I have one question for Omer. When we see this rotational activity named rotor, we are very surprised because we see something that is relatively smooth, like that, we are following the voltage die. And it's nearly impossible to have something so regular in a tissue with a lot of structural abnormality. My question is, how much of optical mapping, providing the phase mapping showing a rotational activity, have been correlated with methods like Natalia or Vadim, just to see how much of these rotors are in reality classical re-entry shown by this way. First in AFib, like in your model of cholinergic AF, but more importantly, possibly for the ventricle. In the ventricle, you publish also this rotor activity. Have you tried to correlate with ultrastructural imaging underneath to the rotor? I mean, the question is, they are rotor shown by phase mapping, but how much of them are in reality some re-entry meandering around the structural tissue?
Well, I'm not sure I understand the intricacies of the question. If the question is whether we see that with different methods, electrical methods, is that what you're asking?
Yeah, in fact, the rotor is seen like in the cosmos, like something that is turning like that, like a spiral. But it's hard to see when we know all this knowledge now about ultrastructural change of conduction, that is really something that is turning this way with a regular front turning. Donc, how much of them you think are in reality re-entry? I mean, really anatomical re-entry turning inside the tissue.
Yeah. Sometimes we describe a picture that is maybe oversimplistic. Obviously, there are multiple scenarios and rotors or re-entries may appear in various ways. So I try to go for the fundamentals. That's why I'm focusing on the singularity point. Although it may be prone to noise and sometimes false positives, it captures the essence of functional reentry, and as long as it's not fully anatomical, when there is an obstacle, you'll always find singularity point. Now, how to detect it, I'm not sure I'm answering exactly what concerns you, but how to detect it may be a whole different story, and well, that's what we're sitting here discussing this. First of all, I am relying on optical mapping, and I try to avoid electrical recordings because the discussion that we're having today basically tells us that we have no real way to address that challenge. The issues of far field and where is the source to the signals that we're seeing is fuzzy. There are maybe theoretical ways to address it, but we're not there yet. So the rotors, the entries appear, as I showed, and as we learn, in many ways, they are not stable. In our experiments, they are rarely stable. In the human, it may be more stable than actually the data that I've shown, because to have reproducible data, we load the heart with carbachol, which may complicate the AF. The AF in the patients may be a little bit more simpler than what we see in our experiments. The frequencies are lower, and they're more simple. I think, I'm not sure. I cannot, we don't have reliable mapping data for AF in the patient. So there are differences between what I see and what probably happens in the patients, but the principle is that all the data that I have suggests that each rotor accelerates when it's existing, and AF depends on a series of rotors that appear to keep that high frequency so new rotors can be formed. I didn't have time to go over all this model. And in my opinion, and again, that may not answer what bothers you, Michelle, you need panoramic mapping. You need to see everywhere, because I don't see any region that is a preferred region. There may be the the pulmonary vein isolation is maybe successful because it increases the amount of boundaries. Of course, it may isolate triggers, but if there are rotors already, the way to stop them, maybe not hitting them straight on the core, but because they drift, they eventually will find a boundary and disappear. These are all thoughts. I don't have strong data for that. We have simulations, but yeah, the mapping is not there yet. The mapping technology is a major limitation. Yeah, I'll stop here. We can discuss that more.
Thank you.
I think we don't have time. I just want to ask Natalia a question, if she's still on the line.
I am here. I am boarding a plane. I am so sorry I cannot be there, but I'm happy to listen to the question. Yes.
Yeah. Hi, Natalia. It's Elad. So I really like your talk, and it really fits everything we know, and it also fits some of the different results we see with different studies about the significance of extra stimulation and pacing from different wavefronts. So I think it really puts it together very, very nicely. Now, just explain to us, what do we need to get it for clinical practice? So there are methods when we do the MRI, and there are some companies that can segment this to us and show us channels and so forth. But ideally, what we want is to combine the simulation that you do on the structure and then provide us not only the structural information, but also the functional information that we can merge with our electroanatomical mapping to guide ablation. So where are we with that? And then one other question to you too is that in the previous session, both Dave and Kevin have shown and wrote about maybe changes, heterogeneity in APD is at least or even more important than activation slowing. Does your model also includes APD? Do you think it's, did you try to incorporate it into your model? Does it make any difference?
So I will start with the second question because it is simpler for me to answer while I'm about to put my bag above here in the overhead compartment. So we do incorporate changes in APD in the gray zone. That has been always feature of all our models. So sometimes we even incorporate it heterogeneously depending on the signal intensity in in the imaging. So generally, that we've always done that. How in the future? What is going to happen? Elad's question was very broad. What do we do with all that? You have all these simulations. We have clinical recordings. How do we combine all that to actually improve what we do with our patients? Am I summarizing correctly, Elad?
Yes.
Yeah, okay. So one thing that I personally would like to do is to incorporate a much larger amount of artificial intelligence in all these approaches. Because right now we can have heterogeneous action potential duration in the gray zone. We can have conduction slowing. We can have all these sophisticated methodologies, but it takes time to calculate everything. So my hope is that we can substitute all that with AI, and that will integrate together clinical recordings, imaging, simulations, and would be able to be part of the clinical workflow, and it's part of on the screen, let's say, when you're playing patients. So I know this seems like a far-fetched vision, but I don't think it is per se, because we really are working on it and we have made some advance. So I'm hoping in a few years, we may achieve that, if you believe me.
Thank you, Natalia, and have a good flight.
Thank you all. Enjoy your meeting.